and this was really suggesting that it, and I think this is widely appreciated now, that it's the receptor tyrosine kinase itself which, when overexpressed, is driving the breast cancer. So, so this family of receptor tyrosine kinases, this is what they look like. So here's a little stick model showing the tyrosine kinase domain inside the cell, these tyrosine residues which become phosphorylated and are so important in downstream signaling that activates cell proliferation. And then there are these 4 domains which make up the extracellular part that binds EGF, or epidermal growth factor, named because it's the key growth factor for epidermal cells, and of course it's the epidermal cells that give rise to most cancers. So it's actually the prototypical family of receptor tyrosine kinases. This is the family that most work has been done on. There are 4 family members, numbers 1 to 4. They have a complex set of ligands, so 7 different ligands, and they're really important for regulating the growth, differentiation and migration of epithelial cells, which of course are so important throughout the body. And as I've already said, aberrant signaling has been implicated in cancer and also in some nervous system disorders. And there's been really interesting research over about the last 10 years into EGF receptors, and some really unexpected activation mechanisms have been uncovered. So the classical textbook model was dimerization induced by the growth factor, just like I told you for VEGF and the VEGF receptor, but that's not the case for EGF receptor, as I'll now show you, or show you in a minute. Okay, so a lot of this has come from structural biology as well. So the structures of all 4 members of the family have now been solved. And for 3 of them, the structures look the same. So this is just the main extracellular, this is just the extracellular region of the receptor tyrosine kinase, so the bit sticking out. And if you imagine, the transmembrane would be here, and then the kinase domain and the tail inside the cell would be down here. So for numbers 1, 3, and 4, they could show that actually these 4 domains folded up into a kind of ball. So you had domain 1, the one at the top, to domain 2, up to domain 3, down to domain 4, and then we'd have our transmembrane here. And very similar for numbers 3 and number 4. But very strikingly, that number 2, this is the one we're going to talk about in terms of breast cancer, it was extended, so it was more like the kind of stick model that everyone had been drawing these kinases as before they realized what the true structure was. So you have domain number 1 here, and then it moves into domain 2, which has this strange sticky out bit, domain 3, and domain 4. And there are various ligands for these. So epidermal growth factor is, I guess, the most well-known one, and that binds only to number 1 in the family. Similarly, TGF-alpha and AR is amphoregulin. You don't really need to know this, but these are also specific to number 1. We've got these NRGs. These are neuroregulins, particularly important in the nervous system. So some only bind to number 4. And then we've got some bispecific ligands that bind to more than 1, so to numbers 1 and to number 4. But what you'll notice about number 2, this rather strange stick-like one that's amplified in these breast cancers, is there isn't actually a known ligand for it. So we still haven't found a ligand that binds to this one. So the ligands only bind to these folded up ones, not to our extended HER2. Oh, and one final wrinkle, again, for the purposes of this, you don't really need to know this, but number 3 is actually inactive as a kinase. So its kinase domain has natural mutations that render it kinase inactive. So 3 of them are active, 1 of them's inactive, and 1 of them has this rather strange extended structure and has no known ligand. And from various structural studies, this was how they figured out that EGF, epidermal growth factor, was actually activating these receptors. So this would be the prototype, number 1. So it exists in this tethered, inactive conformation. And EGF isn't actually dimeric. So it's a monomeric growth factor. And what it will do, it will bind to a region between domains 1 and 3, and it will lock the extracellular part of the receptor tyrosine kinase in an extended conformation. Okay, so it's able to fight its way in there, and once it's bound, it maintains this stick-like extended conformation. So we think that probably these molecules are kind of breathing, there's a bit of flexibility, and maybe they're transiently alternating between this folded up state and this extended state. And the growth factor, when it comes along, it actually locks the extracellular part into this extended conformation. And this little sticky out bit here is called the dimerization arm, and it's able to dimerize by binding to the extended dimerization arm on another activated receptor tyrosine kinase. So the bottom line is the growth factor causes this stick-like conformation. We get the dimerization arm now exposed. It's a sticky dimerization arm, and it can bind to a dimerization arm on another active molecule, and this is how we get dimerization. It's not the classical induction of dimerization by a dimeric growth factor. The growth factor is actually causing dimerization by conformational change. So the kinase inactive one can still be functional, so it can still be activated in the same way. In this case, B3 is the inactive one, and it can still be extended by one of its growth factors, this NRG or neuregulin. And once it's extended, it can then hook up with one of the active members of the family, and when it does this, it will still be able to signal in a slightly different way, because only one of the dimers will have an active kinase domain, but still able to signal. And I guess having a kinase inactive member of the family allows you to get more ways to signal, to increase complexity of the signaling response if it involves this kinase inactive one. And finally, what's going on with our ERB2? So this is the one that's driving breast cancer. Well, this one doesn't need a ligand, okay? It's naturally in this extended conformation, and this is why it's so dangerous, because if you get it overexpressed, it will be able to hook up either with another ERB2 molecule or with, for example, ERB3, the kinase inactive one. And together they can form an active signaling unit. So you really don't want this one overexpressed, because it doesn't need a ligand to get active. But of course, overexpression is what happens in these breast cancers. So the plan was then, people thought, right, if we can make an antibody to this receptor tyrosine kinase that blocks dimerization, this could be a good treatment. And it actually worked spectacularly well. But again, not in the way they thought it would work. So I'll come back to that at the end. So just to give you more information on this HER2, this ERB2, it's certainly not specific to these breast cancer cells. It's got quite wide expression throughout the body, but it's incredibly highly expressed on these tumour cells. So it can be at 100-fold higher levels on the breast cancers than it is on the rest of on other cells in the body. And as I've already mentioned, having amplification of HER2 on your cancer was a really bad thing. But Herceptin was approved in 1998 to treat the breast cancers, and it was actually the first clinically useful antibody to a receptor. And in 2005, a study showed that after surgical removal, if you were treating with this antibody, the relapse 4 years later was reduced by more than half. So this antibody, big success story, and it actually turned a really nasty cancer with very poor prognosis into one that was treatable. So now it's actually, I mean, you never want to have cancer, but it's actually quite good to have HER2 positive cancer because it is treatable with this Herceptin antibody. So it was thought that the way it would work would be to block dimerization, because this was how researchers were showing the molecule worked. It needed to dimerize to start sending a signal and to cause the cell to proliferate. But actually, we now know that that's not the way it works. Instead, the antibody, complicated, but it appears to have two mechanisms. So one is that it actually causes internalization and degradation of the receptor. So it's a way of just removing the receptor from the cell surface. And the other way it works is by antibody-dependent cellular cytotoxicity. So it flags up to the immune system, you know, here I am, I'm an antibody, I'm bound to this tumour cell, come and kill the tumour cell. And the way that works, I'll get to that in the end. Yeah, so I'll now go through a brief history of how this was discovered. So way back in 1989, to make an antibody to this protein, they decided to immunize mice with membranes from fibroblasts overexpressing this HER2 protein to make monoclonal antibodies. And this yielded an antibody that they called 4D5. And they could do Western blots here to show that when they had HER2 expressed in their fibroblasts and made lysates and ran them on a protein gel and did a Western blot, their new antibody would detect HER2, but it wouldn't detect one of the other family members, HER1, so it was specific for HER2, and there are various other controls on that gel. They could do flow cytometry, so this is a technique some of you may know about or have even done, so a way of measuring the expression, the surface expression of an antigen on a cell. So here we have their antibody nicely staining positive to cells overexpressing HER2, but a negative control antibody doesn't stain the cells. And then they went on to do some experiments to test if their antibody was actually going to do what they hoped and inhibit growth of tumours. So here they're simply culturing a breast cancer cell line that has HER2 expressed. This black dotted line, they've treated with their HER2 antibody, and you can see how much slower the cells are growing. When they remove the antibody, then the tumour starts to grow. And then another classic way for assessing anti-cancer reagents is to grow your cells in soft agar. So most cells wouldn't grow in soft agar because they've got nothing to really cling on to. But one of the properties of cancer cells is that they can grow in soft agar, and they form these nice colonies when you grow them in agar. But when you introduce this antibody into the agar, you can see the cells are just not growing, they're not forming these colonies. And then finally, I guess the classic way you do it is you take one of these poor mice, usually an immunosuppressed mice, so you can implant the human tumour cells somewhere in the mouse. Here they use the kidney capsule, or under the kidney capsule, and you can see they have some good effects. So this is showing you the weight of the tumour over a period of time, so 20 days after treatment or 34 days after treatment. The control mice either given phosphate buffered saline or just a control immunoglobulin. You can see their tumours growing nice and big. But when you have treatment with the antibody to HER2, very small tumours. So very nice result showing this antibody was impairing tumour growth. So, yes, so, and this is how This is actually how we think it's working in vivo, so in patients. So clearly in the in vitro studies, the antibody on its own is having some effect, but in vivo we think that the main way the tumour's being killed is by bringing in the immune system. So getting into the clinic, so the antibody was humanized, so to limit the immune response that the patients would make against this mouse antibody. They engineered the antigen recognizing complementarity determining regions into a human IgG molecule to do this. And then they had to go through those same sort of tests again just to check that their humanized antibody still works, still bound HER2, and still worked in these various assays. And the way this humanized antibody seems to work best in a patient is it will bind to the cells. It's the FC portion of the antibody will now act as a flag to bring in immune cells, such as this natural killer cell. The natural killer cell has this FC gamma R3 receptor, which recognizes the FC part of the antibody. This NK cell will then release its cytotoxic granules, and it will reduce the tumour cell to a little puddle. That's the idea. So it's the antibody flagging up to the immune response to enable cells such as NK cells or cytotoxic T cells to kill the tumour, to induce apoptosis. Okay, so the clinical trials then that were done, as you're probably aware, phase 1 is just to look at safety and get an idea of how much of the drug you can give to a person. Phase 2, further safety checks and also to try and see if the drug is doing anything. And then phase 3 of the clinical trial is a big randomized multi-center trial just to see how effective your new drug is in comparison with the gold standards that are available. And so this is a phase 2 study that was published in the late 90s. So 46 patients with HER2 overexpressing tumours, and in a pretty bad way, so lots of metastases, and they've probably already had anti-cancer therapy that has failed. And what they found was that, as you might expect with an antibody therapy, minimal toxicity, because antibodies are often well tolerated. 5 of the 46 patients responded, 4 partial, but 1 went into complete remission. So not everyone was helped, but you've got to remember these were very advanced tumours, and so this was seen as a big success story. So what about combining Herceptin with chemotherapy? So here we're looking at patients who've just received one of these nasty chemotherapeutic agents that inhibits DNA synthesis, or are trastuzumab, that's the Herceptin antibody, plus the chemotherapy. And you can see that patients who have the antibody as well do better. So it takes longer for them to progress their disease, the response rate is better, and their overall survival is better here by 5 months. And this is this is a different cytotoxic drug here, but essentially the same sort of findings. And this was a big story in New England Journal of Medicine, and the eligibility for this was very nasty, progressive, metastatic breast cancer with HER2 overexpressed, who hadn't previously received chemotherapy. So what it does then, it promotes survival, and this may not look very impressive because still most people eventually die, unfortunately, but at least having the combination of the antibody with the chemotherapy extends survival. And I guess some people, they're getting pretty much a complete cure from this. So side effects are generally low. There is some heart dysfunction that has been reported. And just to summarise then this bit, so the aim was to inhibit this HER2 receptor that was driving these breast cancers that had such poor prognosis. It actually appears that it's not working in vivo by blocking dimerization. Instead, probably the main way it works is by bringing in the immune system this antibody-dependent cellular cytotoxicity, so attracting the immune system to kill the cancers which are labelled with this HER2 antibody. And it's also thought that internalisation of the HER2 and degradation of it may also help. Okay, so finally onto the last example. So this is Rituxan, and this is targeting the CD20 antigen on B cells to treat non-Hodgkin's lymphoma, a type of blood cancer, and also more recently been used to treat rheumatoid arthritis, because it's become clear that B cells, normal B cells, have a role in driving that disease. So this is a structural model of this therapeutic antibody called rituximab, binding to this rather tiny little membrane protein called CD20. So CD20 has 4 transmembrane regions. You can probably just make them out in this lipid bilayer, and the extracellular parts are really rather minimal, but nevertheless enough to enable you to make an antibody that recognises that extracellular part. So rituximab itself is actually a chimeric antibody, so it's not humanised. There's a bit more mouse in it than for a humanised antibody. And it recognises this small CD20 protein. And I still think we don't really know what CD20 does. We just know it's very highly expressed on B cells and on certain B cell cancers. But it's been a good target. And so this is the expression of CD20 across the B cell lineage compared to a number of other well-characterized B cell markers. So here we're going from our stem cell all the way through to our plasma cell that produces antibody. And you can see that our CD20 expression starts in the early B cells and then shuts off just before we get to the antibody-producing plasma cell. But a whole bunch of these blood cancers, these leukemias, are expressing very high levels of CD20, which you might expect because they have derived from these various stages of B cell. Okay, and the reason actually it's been so successful is that it's expressed at very high levels on the tumours. Okay, so it's been a good target for an antibody, but also it doesn't get internalised. So, or at least not very much. So with many proteins, you bind an antibody to them and they get internalised into the cell and degraded. Just seems to be a natural consequence of antibody binding. But for CD20, it's very stable, and the antibody will be stuck to the CD20, again, flagging up to the immune system, come and kill me, I'm an antibody, I've bound to a cell. So a brief history then of Rituxan. So this was the first chimeric antibody to be approved by the FDA, and this was for non-Hodgkin lymphoma in 1997. So this is actually the 5th and the 6th most common cause of death for males or females. And then also it's been approved for rheumatoid arthritis because normal B cells are known to have this promoting role in rheumatoid arthritis. And if you hit them with this antibody, okay, it's probably not great to lose a lot of your B cells, but if you've got rheumatoid arthritis, that does relieve a lot of the symptoms. So this was the success story in lymphoma then. So patients who got weekly doses of this antibody, this was a successful treatment for about half of the patients who'd got a relapsed or refractory non-Hodgkin lymphoma. This antibody prolongs life, but it's not actually curative, unfortunately. So for some reason, patients eventually relapse after several years on this treatment. Ultimately, if we combine this with other anti-cancer therapies, maybe a double whammy could end up being effective and could provide a cure. Normal B cells in the body, which express CD20, are also affected, so they get depleted, but these do return to normal levels eventually. And so this is the way this antibody works, by antibody-dependent cellular cytotoxicity. So this is our CD20 molecule on the surface of our lymphoma. When the antibody binds to the CD20, I've already said CD20 tends to not get internalized and it stays on the surface for quite a long time, flagging up to the immune system that there's an antibody bound. Effector cells like natural killer cells or cytotoxic T cells. Actually, I'm not sure cytotoxic T cells will do this, but certainly NK cells have FC receptors and will bind to the FC part of the antibody, and they will release their cytotoxic granules to cause apoptosis and lysis of the cell. And there's another way that this works, so through complement-dependent cytotoxicity. So one of the things that happens to an antibody that's bound to an antigen is the antibody has this complement complex that builds up on its FC portion, and this complement can form this so-called membrane attack complex, which punches holes in the cell and kills them in that way. So that is how this antibody is working. So just to summarize this then, so I've already said it's the first chimeric antibody to be approved, targets this B-cell antigen CD20, and it depletes tumour cells, so extending the life of these patients with non-Hodgkin lymphoma. And as I mentioned, it's now a routine treatment for autoimmune diseases like rheumatoid arthritis that are driven by B-cell function. So that's pretty much it. So what you should know from these lectures, so you should know the strengths and limitations of antibodies as therapeutics, and then also these three stories of Avastin and Herceptin and Rituxan, these three blockbuster antibodies that are anti-cancer antibodies and are really propelling the whole field of therapeutic antibodies because of the big money they're generating. Bit of further reading. I spotted this last one just before I came out. It's got no pictures in it, this last review. I always think it's bad. You've got to have pictures in a review article, haven't you? But at least it's a very up-to-date kind of state-of-the-art of all the anti-cancer antibodies out there. So by reading this, you'll probably get a slightly updated version of things than what I've told you. And then these other two ones do have nice pictures, so I'd recommend these two if you like pictures, but they're not quite as up-to-date. So that's it. Thank you very much for listening.